Hi students, welcome back to online lectures. Now we are moving on to Unit 4, Topic 1. In this we are going to discuss about regulation of gene expression, clustered genes and Operon concepts, negative and positive control of Lactose. So in this unit we are totally going to discuss about regulation of gene expression, what is the function of the gene, and how the gene is expressed and how the product is synthesized and what are the regulatory mechanisms to control the gene expression. In this topic, we are going to discuss about the expression of the lactose genes in which we should study negative and positive control of. Coming to gene expression and regulation. Organisms adapt to environmental changes by altering gene expression. The regulation of gene expression of gene is necessary for the growth, development, differentiation and the existence of the organism. What is gene expression? So to understand the function of the gene, how a product is synthesized, we must know what is the expression of a gene. It is the process by which information from a gene is used in the synthesis of a functional gene product. These products are often proteins. So what are the products of the gene? They are often proteins. But in non-protein coding genes, such as rRNA genes or tRNA genes, the product is functional RNA. So what does it mean? Whereas for the non-protein coding genes such as rRNA genes or tRNA genes, the product is a functional RNA. Types of the genes based on their functions. They are of inducible genes and constitutive genes. What are the inducible genes? The expression of the inducible gene increased in response to an inducer. So inducible genes, that means expression of the genes are enhanced in the presence of certain molecules. They are known as inducer molecules. Some proteins produced by Escherichia coli. Example, beta-galactosidase are said to be inducible. What is that? Example, beta-galactosidase are said to be inducible because they are only produced in significant amounts when a specific inducer lactose is present. What does it mean? So, beta-galactosidase encoding gene is expressed when the inducer molecule lactose is present. In the absence of the lactose, beta-galactosidase gene do not express. Another example is tryptophan pyrrolase of liver is induced by tryptophan. So another example is tryptophan pyrrolase of the liver is induced by the molecule tryptophan. These are known as inducer molecules involved in the expression of the genes. Such genes are called inducible. Coming to constitutive genes. The constitutive genes are expressed at more or less constant rate in almost all the cells and they are not subjected to regulation. So what does it mean? Constitutive genes are expressed at more or less constant rate. In almost all the cells they are present, they are constantly expressed and they are not subjected to regulation. The products of these genes are required all the time in cells. Example, enzymes of citric acid cycle. Go back. నువ్వు వెనక్కి వెళ్ళు సాత్వి. Positive regulation. When the expression of gene is quantitatively increased by the presence of specific regulatory element is known as positive regulation. So what is positive regulation? Genetic expression is enhanced in the presence of regulatory element known as positive regulation. Here the element modulating positive regulation is known as activator or positive regulator. Here the molecules which enhances the expression of the gene, which induces the expression of the gene, they are known as activators or positive regulators. The mechanism is called. Next. Coming to negative regulation. We discussed positive regulation when inducer molecules, activators, positive regulators, which enhances the expression of the gene are known as positive regulators. Negative regulation is when the expression of the gene is diminished or depressed by the presence of specific regulatory element. So what does it mean? Because of the presence of certain molecules, the genetic expression is decreased or diminished, that means stopped. Such elements are called negative regulatory elements, and the regulation is called negative regulation. The molecules are known as repressors. The molecules are known as repressors. So activators are positive regulation. Repressors induces negative regulation. So now we are going to discuss about gene expression in prokaryotes. So we are going to study an example of a prokaryotic cell in which how the genes are expressed using the positive regulation and negative regulation mechanisms. That means what are the activator molecules, what are the repressor molecules involved in the functioning of the genes. So here the concepts are known as Operon concept. The concept of operon was first introduced by Jacob and Monod in 1961. So, who explained the operon concept? Jacob and Monod in 1961. Operon is defined as a segment of DNA strand. So, what is operon? It is a segment of the DNA strand consisting of, what they consist of? Structural genes. What are the structural genes? Cluster of several structural genes which carries the codons which can be translated into proteins. So what is structural gene? A cluster of several structural genes which carries codons and which can be translated into proteins. They are known as structural genes which encodes proteins. Next type of the genes are operator genes. And one operator gene which has an overall control over the process of translation. That means to express the structural genes, they require the operator genes to be functioned. So overall control of the structural gene expression is achieved by the operator genes. So operator means which controls the process of the expression of the in the operon. So one type structural genes, another type operator genes, and third type of the genes are regulator genes. So operon is made up of structural genes, operator genes and regulator genes. A third gene called regulator gene which is located sometimes at a distance from the operator gene on the same DNA strand. So what is regulator gene? Which is involved in the regulation of the gene expression which is found very distance from the operator gene. Regulator gene transcribes mRNA which synthesizes repressor protein. So what is the function of the regulator gene? Which is expressed and synthesizes a repressor protein which regulates the transcription of the subsequent region of the operon. That means to express the structural genes or to depress the structural genes, regulatory proteins plays important role. So that gene is found distance from the promoter region. So what is the promoter region which is situated between the operator gene and the regulator gene? So P region, promoter region is found between the operator region and regulator region. Now you can see the picture of the Lactose here and how structural genes, operator genes, promoter genes and regulatory genes are organized on the same stretch of the DNA strand. Here you can see what are the structural genes, Z gene, Y gene and A. These genes are known as structural genes. And upstream to these region, operator region is present. To the operator upstream, promoter site is present. To the promoter site upstream, regulator gene is found. See the sequence, regulator genes, promoter site, operator site. So, promoter is found between the operator and regulator gene and followed by structural genes. So, to express the structural genes, first the operator should control all these structural genes. What is the function of regulator genes? They express and synthesizes repressor. These repressor molecules able to control the operator regions. Lactose operon. We are going to discuss now lactose operon in Escherichia coli. The lac operon is an inducible catabolic operon of E. coli. Inducible catabolic operon means this operon is expressed in the presence of certain inducible molecules. It consists analysis of structural genes. What are the structural genes? Z gene, Y gene and A gene. So Z gene encodes beta-galactosidase. Y gene encodes galactoside permease. And A gene encodes thiogalactoside transestylase. So these are the structural genes of the lactose operon. So Z encodes beta-galactosidase, Y encodes galactoside permease, A encodes thiogalactoside. Now we will see the functions of the genes in this session. Beta-galactosidase, this is the enzyme which is expressed by the gene Z. Hydrolyzes lactose. This is also known as beta-galactoside to galactose and glucose. Beta-galactosidase enzyme hydrolyzes the lactose to galactose and glucose. Next one, permease. This is responsible for the transport of lactose into the cell. So when Escherichia coli is growing in a medium containing lactose, so this permease situated in the cell membrane and allows the entry of the lactose into the cell. So after entering into the cell, this is splitted into galactose and glucose by beta-galactosidase. And next one is acetylase, which is coded by A gene, and mostly the function is acetylation of the lactose. So the structural genes Z, Y, A transcribe. So the genes expressed nothing but they transcribe to form a single large mRNA. So the three genes are expressed at once into a form of large mRNA with three independent translation units. That means Z can independently translate it, Y can independently, likewise A can independently translate it. And for the synthesis of three distinct types of the enzyme, nothing but beta-galactosidase, permease and acetylase. Such a mRNA coding more than one protein is called polycistronic mRNA. That means all the three structural genes are transcribed at once. So such large mRNA is known as polycistronic mRNA. So which is characteristic in prokaryotes. So in the next session, we will see the mechanism of the regulation. Thank you.